This video will cover the structure and function of prokaryotic cells, focusing on bacteria. We will describe the structure, discuss the functions of various components, list morphological types, and differentiate between gram-positive and gram-negative bacteria, including the clinical significance of their cell wall differences. The basic components of a bacterial structure can be divided into three architectural regions: the cell envelope, the cytoplasmic region, and the appendages. The cell envelope includes the capsule, cell wall, and plasma membrane. The cytoplasm contains the DNA, ribosomes, and various inclusions. Appendages are structures like flagella and fimbriae. This diagram illustrates the structure of a typical bacterium, including the capsule, cell wall, and plasma membrane, which form the envelope. Inside, you see the nucleoid, ribosomes, and depot substances. Appendages like the flagella and attachment pili are also visible. The outermost covering of bacteria can be a loose slime layer that is easily washed off, or a thick, structured capsule that is not readily removed. The image also shows a microscopic view of encapsulated bacteria. The capsule is the outermost covering, usually made of polysaccharide. This layer protects the bacterial cell from phagocytosis and antibacterial agents. Pathogenic bacteria with a capsule, such as Streptococcus pneumoniae, resist engulfment by leukocytes. Biofilms consist of bacteria living in a matrix of polysaccharide slime or glycocalyx that they secrete. This aids in adherence to damaged tissues and plastic surfaces. Biofilms offer advantages, making the bacteria less susceptible to drugs and phagocytosis. Examples include dental plaques and Pseudomonas spp. Biofilms on catheters. The cell wall is composed of peptidoglycan and maintains the overall shape of the bacterial cell, such as coccus, bacillus, or spirillum. It also provides protection from the environment. An exception is mycoplasma, a bacteria with no cell wall and thus no definite shape. Peptidoglycan, or murine, is a vast polymer of interlocking chains of identical monomers. Each peptidoglycan monomer consists of two amino sugars, N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, with a tetrapeptide chain extending from the NAM. Gram staining, developed by Hans Christian Gram, is a widely used technique in diagnostic bacteriology. It differentiates bacteria into two main groups, gram-positive and gram-negative bacteria, based on their cell wall structure. The fundamental difference between gram-positive and gram-negative bacteria lies in their cell wall structure. Gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thinner peptidoglycan layer and an additional outer membrane containing lipopolysaccharide, LPS. Bacteria have several morphological types. Cocci are spherical, such as Staphylococcus and Streptococcus. Bacilli are rod-shaped, like Escherichia coli. Coccobacilli are intermediate, and spirochaetes, like Treponema pallidum, are spiral-shaped. The cell wall components are major determinants of virulence. Endotoxin in gram-negative bacteria is toxic. Peptidoglycan, LPS, and teichoic acids can induce inflammation. Teichoic acids in gram-positive and O-specific polysaccharide in gram-negative bacteria act as adhesins. Endotoxin is a powerful immune stimulus that activates immune and clotting pathways. High levels can cause septic or endotoxin shock, leading to fever, vascular collapse, multi-organ failure, and death. Examples include septicemia with Neisseria meningitidis and Escherichia coli. A key comparison shows gram-positive bacteria have a thick peptidoglycan layer and are more susceptible to penicillin. Gram-negative bacteria have a thin peptidoglycan layer, an outer membrane, and are more resistant to antibiotics due to the presence of lipopolysaccharide and porins. The cell wall is clinically significant because it helps in diagnosis, distinguishing between gram-positive and gram-negative bacteria. This information aids in the early initiation of treatment and the selection of appropriate antibiotics, as gram-negative bacteria are generally more resistant. The plasma membrane is a semi-permeable lipid bilayer with embedded proteins. It is crucial for the transport of ions, nutrients, and waste. It also serves as a site for metabolic processes, including respiration and the synthesis of lipids and the cell wall. The plasma membrane plays a role in pathogenesis by maintaining cellular integrity, secreting toxins, and contributing to resistance against antimicrobial agents. It also facilitates a tactic response and senses environmental signals to activate virulence genes. The cytoplasmic matrix contains the nucleoid, where the DNA is centrally located and lacks a nuclear membrane. Extrachromosomal DNA is found in plasmids. Ribosomes, composed of 50S and 30S subunits, translate mRNA into protein sequences. The cytoplasmic matrix also contains storage granules, which reserve nutrients like glycogen, lipids, and polyphosphate. Metachromatic or volutin granules, which are phosphate reserves, are notably seen in Corynebacterium diphtheriae. Pili, or fimbriae, are hollow, hair-like protein structures that allow bacteria to attach to other cells. They are antigenic, but antibodies against pilin are not always protective. The two main types are common pili/fimbriae and sex pili. Common pili or fimbriae are major determinants of bacterial virulence, enabling pathogens to attach to tissues and resist phagocytic attack. They are antigenic, and secretory IgA antibodies can block colonization. They are present in gram-negative and some gram-positive bacteria. Flagella are long appendages used for locomotion. They rotate using a motor under the cytoplasmic membrane. Bacteria can have varying numbers of flagella in different positions, enabling them to move within their environment. Flagella aid in tactic movement, such as chemotaxis, moving towards nutrients or away from harmful substances, and aerotaxis, moving based on oxygen levels. They are a determinant of virulence, for example, helping Vibrio cholerae avoid being flushed from the gut. Flagella are also antigenic. Bacterial spores are highly resistant, dormant stages formed under unfavorable conditions. They are resistant to desiccation, heat, and chemicals due to their dehydrated state and the presence of calcium dipicolinate. Spores germinate to produce a single vegetative cell when conditions become favorable. Two major bacterial genera produce spores: Bacillus, an aerobic gram-positive rod with non-bulging spores, and Clostridium, an anaerobic gram-positive rod with bulging spores. Examples include Bacillus anthracis and Clostridium tetani.